Hi everyone, I am Dr. C. Sulprani and MD and I am going to teach medical biochemistry. Before I start the topic, please like, share and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with fatty liver. In today's session, we will continue with fatty liver and lipotropic factors where we deal with their causes. Then we have metabolism of ketone bodies, where there are ketogenesis and ketolysis respectively. Good morning, everyone. Let us begin the topic. Fatty liver and lipotropic factors. The number one cause of fatty liver is excessive mobilization of fat. The capacity of liver to take up the free fatty acids from blood far exceeds its capacity for excretion as VLDL. So fatty liver can occur in diabetes mellitus and starvation due to increased lipolysis in adipose tissue. Here we can see causes of fatty liver. It can be due to free fatty acids and blood glucose entering into the liver, or it can be due to triacylglycerol moving from liver to muscle, or it can be due to oxidation of triacylglycerol. Second important cause is excess calorie intake. Excess calories, either in the form of carbohydrates or as fats, are deposited as fat. Hence, obesity may be accompanied by fatty liver. Third important cause is toxic injury to the liver. In toxic injury to the liver, due to the poisoning of compounds like carbon tetrachloride and arsenic, the capacity of the synthesized VLDL is affected. Hence, fatty acid infiltration of liver occurs. In protein calorie malnutrition, amino acids required to synthesize apoproteins may be lacking. Occurrence of fatty liver in protein energy malnutrition may be due to defective apoprotein synthesis. Hepatitis B virus infection reduces the function of hepatic cells. Fourth important cause is alcoholism. It is the most common cause of fatty liver and cirrhosis in India. Alcohol is oxidized to acetaldehyde. This reaction produces increased quantities of NADH, which converts oxaloacetate to marate. As the availability of oxaloacetate is decreased, the oxidation of acetyl-CoA through citric acid cycle is decreased. So fatty acid accumulates, leading to triacylglycerol deposit in liver. Fifth important cause is non-alcoholic fatty liver disease and non-alcoholic steatohepatitis. Non-alcoholic fatty liver disease is the most common cause of disease in liver in India, where fat is accumulated in hepatocytes. High fat diet and uncontrolled diabetes mellitus are the most common causes. As the disease progresses, inflammatory reaction occurs, which is then termed as non-alcoholic steatohepatitis. Sixth important cause is cirrhosis. Fat molecules infiltrate the cytoplasm of the cell, that is, fatty filtration occurs. These are seen as fat droplets, which are merged together to show that most of the cytoplasm becomes laden with fat. The nucleus is pushed to a side of a cell. Nucleus disintegrates, that is, karyorrhexis occurs, and ultimately the hepatic cells is lyzed. As the healing process, the fibrous tissue is laid down, causing fibrosis of liver, otherwise known as cirrhosis of liver. Liver function tests show abnormal values when chronic liver damage occurs. Coming to lipotropic factors, deficiency of these factors may result in fatty liver. They can afford protection against the development of fatty liver. Number one is choline. Feeding of choline has been able to reverse fatty acid changes in animals. Number two is lecithin and methionine. They help in synthesis of apoprotein and choline formation. The deficiency of methyl group for carnitine synthesis may also hinder fatty acid oxidation. Number 3 is vitamin E and selenium. They give protection due to the antioxidant effect. Number 4 is omega 3 fatty acids, which is present in marine oils and has a protective effect against fatty liver. Coming to metabolism of ketone bodies. Fat is burned under the fire of carbohydrates. The acetyl-CoA from fatty acids can enter and get oxidized in TCA cycle only when carbohydrates are available. During starvation and diabetes mellitus, the acetyl-CoA takes the alternate fate of formation of ketone bodies. Coming to the first step, ketogenesis. Acetoacetate is the primary ketone body, while beta-hydroxybutyrate and acetone are secondary ketone bodies. They are synthesized exclusively by liver mitochondria. First step is condensation. Two molecules of acetyl-CoA are condensed to form acetoacetyl-CoA. In this diagram, we can see the first step, where two molecules of acetyl-CoA combine to form acetoacetyl-CoA in the presence of enzyme acetoacetyl-CoA synthase. Second step is production of HMG-CoA. One more acetyl-CoA plus acetoacetyl-CoA gives HMG-CoA, that is beta-hydroxy beta-methyl glutaryl-CoA. The enzyme is HMG-CoA synthase. The mitochondrial HMG-CoA is used for ketogenesis, while cytosolic fraction is used for cholesterol synthesis. In this diagram, we can see that acetoacetyl-CoA plus acetyl-CoA together give beta-hydroxy beta-methyl glutaryl-CoA and the enzyme HMG-CoA synthase. Third step is lysis. Then HMG-CoA is lysed to acetoacetate. Acetoacetate may also be formed by the degradation of carbon skeleton of ketogenic amino acids like leucine, lysine, phenylalanine, and tyrosine. HMG-CoA lyase is present only in liver. Here we can see that beta-hydroxy beta-methyl glutaryl-CoA is converted into acetoacetate plus acetyl-CoA in the presence of enzyme HMG-CoA lyase. Step 4 is reduction. Beta-hydroxybutyrate is formed by reduction of acetoacetate. Ratio between acetoacetate and beta-hydroxybutyrate is decided by the cellular NAD to NADH to NADH ratio. Fifth step is spontaneous decarboxylation, where acetone is formed. Here we can see acetoacetate plus acetyl-CoA gives beta-hydroxybutyrate with the help of enzyme dehydrogenase, or else it can form a spontaneous and non-enzymatic method to form acetone. Coming to the step B, ketolysis. The ketone bodies are formed in the liver, but they are utilized by extrahepatic tissues. The heart muscle and renal cortex prefer the ketone bodies to glucose as fuel. Tissues like skeletal muscle and brain can also utilize ketone bodies as alternate sources of energy if glucose is not available. Acetoacetate is activated to acetoacetyl-CoA by thiophorase enzyme. It is written as acetoacetate plus succinyl-CoA gives acetoacetyl-CoA plus succinate in the presence of enzyme thiophorase. Then acetoacetyl-CoA enters the beta-oxidant pathway to produce energy. Here we can see formation, utilization and excretion of ketone bodies. First, free fatty acids present in the blood enter in the liver and form acetyl-CoA. Then in the liver, this acetyl-CoA is converted to ketone bodies. Then it can be uh, acetone in the breath or ketone bodies in the urine. And it can also form acetyl-CoA, and through citric acid, the carbon dioxide is released. Coming to ketosis. First, normally the rate of synthesis of ketone bodies by the liver is such that they can easily metabolized by the extrahepatic tissues. Hence, the blood level of ketone bodies is less than 1 milligram per deciliter. Only only traces are excreted in the urine, which are not detectable by usual tests. But when the rate of synthesis increases, the ability of extrahepatic tissues to utilize them there also increase. There will be accumulation of ketone bodies in the blood. This leads to ketonemia, the uh, uh, excretion of in urine, that is ketonuria, and smell of acetone in breath. All these three together constitute the condition known as ketosis. Coming to causes for ketosis. The first important cause is diabetes mellitus. Uncontrolled diabetes mellitus is the most common cause for ketosis. Even though glucose is in plenty, the decreased insulin causes increased lipolysis and more fatty acids are released into the circulation. Oxidation of these fatty acids increase the fatty acid acetyl CoA pool. Increased gluconeogenesis restricts the oxidation of acetyl-CoA by TCA cycle since availability of oxaloacetate is decreased. Second cause can be starvation. In starvation, dietary supply of glucose is decreased. Availability of oxaloacetate is channeled to gluconeogenesis. The increased rate of lipolysis is to provide an alternate source of fuel. The excess acetyl-CoA is converted into ketone bodies. The high glucagon level favors ketogenesis. The brain derives 60 to 75% of energy from the ketone bodies under conditions of prolonged starvation. Hyperemesis, that is by vomiting in early pregnancy, may also lead to starvation like conditions and may lead to ketosis. Here ends a session. Kindly like, share, and subscribe the channel. Thank you.